Mitochondria are the primary energy generator of the cell and have unique organelles that maintain their own DNA, mtDNA. In human genetics, human mitochondrial DNA haplogroups are collections of similar haplotypes defined by combinations of single nuclear polymorphism (SNPs) in mtDNA inherited from a common ancestor. Mitochondrial DNA is passed down through cytoplasmic inheritance, where, upon fertilization, the paternal mitochondria are degraded, leaving only the maternal mitochondria regardless of the offspring's sex. This characteristic of mitochondrial inheritance allows geneticists to track the movement and divergence of different haplogroups from female lineages. Haplogroups are used to represent the major branch points on the mitochondrial phylogenetic tree. Understanding this mechanism of inheritance has helped population geneticists trace the matrilineal inheritance of modern humans back to human origins in Africa, and the subsequent spread around the globe. The letter names of the haplogroups, not just mitochondrial DNA haplogroups, run from A to Z. As haplogroups were named in the order of their discovery, the alphabetical ordering does not have any meaning in terms of actual genetic relationships. The hypothetical woman at the root of all these groups, meaning just the mitochondrial DNA haplogroups, is the matrilineal most recent common ancestor, MRCA, for all currently living humans. She is commonly called mitochondrial Eve. The rate at which mitochondrial DNA mutates is known as the mitochondrial molecular clock. It is an area of ongoing research, with one study reporting one mutation per 8,000 years. Recent research has shown that mitochondrial DNA haplogroups can influence risk for various diseases and cancers. Mitochondrial haplotypes vary based on latitude. If you have ever had genetic testing done, you can take your raw data file and run it through dna.jameslik.com to get your haplotype. Otherwise, trace your maternal line back as far as you can to estimate. Haplotypes H, J, T, U, UK, and V are Scandinavian haplotypes. These are among the most uncoupled haplotypes. Uncoupled mitochondria are called as such because they have uncoupled the flow of electrons from the production of ATP, and in doing so, are able to dissipate heat, infrared light, readily. This is important in the face of the extreme cold, low sun conditions in Scandinavia in the winter. In the process of making infrared light, mitochondrial metabolism also releases UV biophotons. So, when cold stimulates the mitochondria, both the production of infrared and UV light is being stimulated. In other words, IR and UV light are so critical for the body that when IR and UV light from sunlight are scarce, i.e. it's winter time, the body makes more of its own. Uncoupled mitochondria evolved to deal with cold, dark winters and warmer longer days in the summer. In contrast, the most coupled haplotypes are L0 and L1. These haplotypes are the most primordial for our species, stemming from the cradle of humanity in southwest Africa. All other haplotypes evolved from L0. Mitochondria with L haplotypes are expecting to receive an abundance of high-quality, high UV index, sunlight year-round, and are not naturally equipped for dealing with low light and prolonged cold temperatures. Because the uncoupled haplotypes evolved more recently, they retain plasticity, that is, memory of their past. What is meant by this is that uncoupled mitochondria are able to shape-shift and do well in more equatorial conditions over time. In contrast, the primordial coupled mitochondria do not have the ability to fully adapt to high-latitude regions with cold, dark winters.
So, this may be a primary reason why Black and brown populations in the US face serious health disparities. Yes, there are socioeconomic factors that play a role, but the relatively high latitude of most of the continental US means that these individuals do not receive adequate light quality for almost half of the year to optimally support their mitochondria. Having said that, even folks with uncoupled mitochondria are suffering because of the normalization of indoor lifestyles, also known as no IR light, no UV light, and no cold temperatures. This is a perfect storm for mitochondrial dysfunction and the chronic diseases that stem from it. Note that the other haplotypes indicated on the graphic are intermediate, meaning that they're not fully uncoupled nor fully coupled. If you have one of these haplotypes, determine which side of the spectrum your mito fall on by finding the latitude associated with that haplotype. For example, haplotypes I, W, and X are on the uncoupled side, while M and N are more coupled. The haplotype H, specifically H2A2A1, meaning that mitochondria are very uncoupled and can do well getting cold for part of the year and getting sun for the other part of the year. If you have uncoupled mitochondria and are living away from the equator, getting cold is a non-negotiable. If you have coupled mito and live above 30 degrees latitude, consider purchasing a Spurti vitamin D light or a tanning salon membership to help offset the mismatch. I recommend trying to find a hybrid tanning bed that has both red and UV light. We are meant to always receive UV with red plus IR light.